everyone. Welcome to the Biorigin Webinar Series. I hope all of you are doing well. I would like to thank you very much for your time and availability to be here today. I am Thaila Cristina Putarov, the Global Technical Manager for Pet Food at Biorigin. Today we are going to have a presentation about the benefits of beta-glucans 1316 for pets with where the speaker will be Professor Aulus. But first of all, I would like to make a short introduction about our company. Talking some words about Biorigin, our company is a Brazilian company founded in 2003. And since then, we use scientific knowledge and biotechnology to improve the health and welfare of humans and animals. Through our biotechnological expertise, we produce natural ingredients for human and animal nutrition. We provide natural ingredients that contributes to palatability, shelf life extension. We also produce alternative protein sources with a very interesting amino acid profile and some other ingredients that can promote health in pets and animal production too. One of our goals is to add value to animals and humans' life, and as a consequence, we add value to our clients' products. Biorigin is part of a bigger group called Zillar Group that farms sugarcane to produce ethanol and sugar. And due to this, we have a great know-how in the fermentation process, and all of this knowledge is also applied for Biorigin products. We have 4 production units. 3 of them are located in São Paulo and have the focus to produce ingredients derived from yeasts. And 1 unit is located in the United States of America, and the main activity there is to produce fermented ingredients for beneficial bacteria. Biorigin has more than 500 customers distributed in 60 countries. And to improve our relationship with our clients, we have offices, warehouse, and sales representatives in several places. Here in Brazil, we also have our Center of Research and Development, where all the time we are developing new technologies, products, and give some support to our clients. Our company is constantly investing in our people and structure. The company increased fourfold its production capacity since 2003. The feed segment focuses to solve problems in the animal production chain and also improve the life quality of pets. And because of this, its primary concept is to work with the functionality of ingredients. For this, we have special yeast strains that has an important role on developing of natural ingredients, such as beta-glucans, mannans, high-quality proteins, peptides, and nucleotides. Through these ingredients, we improve diet palatability and boost the natural defenses of animals during the challenge periods in their lives. Here is a quick overview of our portfolio for all species. We have here Macrogar, that's our purified beta-glucan, and has a lot of benefits for dogs and cats, as we are going to see later. We have also 2 sources of prebiotics of mannanoligosaccharides, where one is a classical one, and the other ones we have soluble mannanas. Selemax is our organic selenium source. Protemic is our mycotoxin adsorbent. Prowin, a natural prebiotic for ruminants. Biotide Extra is our nucleotide sources. Nutricell and Brucell are our inactive dry yeasts. Nutricell is obtained from sugarcane, while Brucell is obtained from the brewer's yeast. High-cell is our autolyzed yeast and has a high level of protein. And if you want to know about all of these products, please let us know. Today, our speaker is going to be Dr. Aulus Cavalieri Carciofi. He graduated in veterinary medicine and specialized in nutrition and nutrition diseases of dogs and cats. He's a professor at São Paulo State University in Brazil, and he is the supervisor of a program of residence in companion animal clinical nutrition. He is the coordinator of the Laboratory of Research in Nutrition and Nutritional Diseases of Dogs and Cats. He is also a faculty member at Kansas State University in US, and he is the vice president of the European Society of Veterinary and Comparative Nutrition. Today, he's going to speak about the benefits of beta-glucans 1,3 and 1,6 for pets. And it's a pleasure to have you here with us today. I wish you a good lecture. Dear all, if you have any questions during the presentation, please just write down on the in the Q&A box, and we are going to be glad to answer at the end of the presentation. Professor Aulus, please, it's your time, and I hope that you have a wonderful lecture.
Hello, it's a big pleasure to be here today to talk to you about beta-glucan for pets. I'd like to thank Biorigin to invite me to be here, and especially I'd like to thank Thaila and Biorigin for the long partnership they have with our laboratory. So Biorigin along these years funded many PhD and master science students, and this was very important for us during this time. And today they invited me to talk about beta-glucan. I'd like to first start what drives an owner to select a pet food. And with the humanization that we experience these days, owners choose food as it was for themselves. So dog and cat owners are not nutritionist experts. So they understand what they want from themselves and they imagine that that was important and also good for the animals. What wants the industry? The industry wants to sell super premium products because these super premium products are more profitable for them. And what was, in my view, the first 4 points important in this relationship, the owner and the companies? First, people expect superior nutritional quality products when we are talking about super premium foods. Also, food safety is very important. And also, it's necessary that the company have the ability to communicate all of this to customers. And also, it's important to remember that this is not science. Commercial classification is based on technical principles, but it's not science, it's more commercial, as the name. And regarding the nutritional quality, they need to be nutritional balance. The raw material quality is very important, and also they need to have a correct processing, extrusion, drying. Food safety is maybe the most challenging part of this picture, and it includes raw material and final product quality control. And also, it's important to have product that was evaluated or certified in animals in scientific studies. And this is maybe the 2 points more important to us in this discussion today, because we are not talking about marketing, that's not my expertise. And also it's interesting to remember that companies like Biorigin can help you in developing your super premium products when they have studies correctly conducted in dogs and cats and published papers that support the claims for the use of their products. And how to develop a product with the additional benefits to health. And the point that that we discussed is it must be complete and balanced, and this is mandatory. However, all competitors also claim or have products like this. And is maybe, in my opinion, the main challenge to have these complete and balanced products. So good amino acid profile is very important. And not only the protein per se need to be considered, but the amino acids are more important. And tryptophan and methionine is maybe the most challenge to balance for dogs, and phenylalanine and tyrosine and tryptophan for cats. Also, it's very important that the fatty acid profile of the diet is good, with special attention to EPA and DHA. Control phosphorus excess is also a challenge in formulation superior quality foods because high ash animal meals or the use of sodium phosphates in some circumstances lead to high phosphorus in diet. And new data pointed that this is not very safe for kidney health. And thinking in long-term health, especially in cats, is very important to consider to restrict or to use just the right amount of phosphorus. Also, I suggest to formulate diet based on dietary fiber and starch because sometimes we see companies that use crude fiber and nitrogen-free extract. This is not a correct approach to understand the carbohydrates of the food because crude fiber solubilize a lot of fiber that is incorrectly included in the nitrogen-free extracted, and this turn very difficult to understand the real digestibility and the real impact of the diet on the gut. And also I consider it a challenge to achieve a good macro element balance. So calcium, phosphorus, sodium, potassium, chlorate, and magnesium. These 6 elements and also sulfur from the sulfur amino acids must be integrated together and the food-based excess need to be calculated. This is very important for the acid-base metabolism and the urine pH in both dogs and cats. Food safety may be the big challenge here. And the main points that I consider important to have the superior quality, super premium food is to achieve a good oxidative stability, no salmonella, guarantee no salmonella or pathogen microorganisms, a proper control of biogenic amines in animal byproducts, adequate control of mycotoxins in vegetable byproducts, control of heavy metals in all materials coming from vitamins, minerals, and also animal proteins might have some heavy metals. We have studies showing that. And also the control of foreign materials like metal, plastic, dirtiness, and this is important when considering raw material cleaning now. And all of these 2 points, food safety and superior balance in nutritional balance are considered very, very important. But maybe food safety is the most challenge that the owners is looking for when they are looking for a super premium food. And a third point that we can consider as a complementary approach is the use of nutraceuticals. So they also is important because owners are looking for something more than just a complete and balanced nutrition. And this is an approach that you can try to improve your diet and the perception of the quality of your diet in the marketing. And nutraceuticals are an alternative here. And nutraceuticals are difficult to define. So in one definition, they can be considered food supplements with concentrated form of bioactive food components presented separately from the food matrix. So purified, and used to increase the health performance of an animal. In another definition, a product obtained from natural food sources that shows health benefits besides their nutritional value, so more than the nutrients. And in another one, a product that are more than a food but less than a pharmaceutical. So nutraceutical is a word built to join together these two concepts, the nutrition and the pharmaceutical. And currently we don't have international accepted definitions. And how are the potential nutraceuticals or complementary approach that we can use to balance or to create the super premium foods? For example, we have in the marketing, and we have published studies supporting these claims of high dosages of EPA and DHA. They might be important for renal and cardiac disease, joint health, cognition, skin health during inflammatory response. Also, elevated dosage of vitamin E, for example, and other antioxidants as beta-carotene, lycopene, and this can climb better immunity and better antioxidant protection. Another example is curcumin. Curcumin have anti-inflammatory, antioxidants, better brain function, cancer, arthritis prevention. So you have several publications in animal models, in humans, that we can translate to dogs and cats on the use of curcumin. And what is very well used in Brazil is prebiotics. I don't know in other parts of the world, but here in Brazil, most of the pet food formulations include prebiotics. And prebiotics like fruit oligosaccharides, GOS, inulin, mannan oligosaccharides might help in creating this health appealing in your product. And we use prebiotics to modify the composition and metabolic activity of the gut microbiota. And then we need to communicate that to owners and see if how that will impact the health of the animals. Just to exemplify this approach, we did an experiment for Biorigin, and we published last year about the effect of the solubility of the yeast cell wall preparation in their prebiotic effects in dogs. So we tested in this study two preparations, and here's the chemical composition of the yeast cell wall preparations that we tested. So the first have a little less total mannan oligosaccharides, but what is important, it was a conventional with insoluble mannan oligosaccharides. And the second preparation have more soluble. So we see that it's 10 times more soluble mannan oligosaccharides in this preparation. And this was the main difference between the two ingredients. And then we fed this in the diet for dogs, health dogs. And what we saw is the soluble compounds induced a greater production of butyrate. And butyrate is a short-chain fatty acid, very important for good health. Also, it induced a lower lactate because we can explain that this lactate was bioconverted in butyrate, showing this change in the metabolic pathway of the microbiota. Also, we saw some changes or tendencies of changes in biogenic amines. So putrescine was increased in dogs fed the soluble yeast cell wall, and spermidine tend to be higher. And we know that putrescine usually is considered undesirable, but they are undesirable when is in high levels in animal byproducts that was incorrectly produced. But our biogenic amines have some physiological actions, and in fact, endogenous production of putrescine, and especially spermidine, help many cellular functions and can be considered very important for the gut cell metabolism. Also, the intake, especially of this more insoluble preparation of yeast cell wall, increased, uh, shows some changes in, oh no, sorry, I confused here. It was the soluble preparation. It reduced the interleukin 6, that is a pro-inflammatory cytokines. And also, it tended to decrease TNF alpha, that is also another pro-inflammatory cytokines. So not only the gut microbiota was changed, the gut microbiota metabolism, but also the consequence was a lower inflammation in the body. And we can argue that this is very interesting for a dog. Also, we measure some parameters in immunology, and monocyte, peripheral monocytes of these dogs exhibited a greater capacity of phagocytosis. And phagocytosis is a very important innate immunity that remove particles or microorganisms that invade the animal. And this is very important to the health of the animal. So we can see here that the consumption of a diet supplemented with yeast cell wall increased phagocytosis activity in peripheral blood of dogs. So this was an example of how we can use these special ingredients to create more than just or only complete and balanced food. For sure, they need to be complete and balanced. Otherwise, even the immunological response cannot be adequate. But sometimes we can complement and differentiate your product, inducing this new health appealing for the animals. So, and coming now in our conversation to the beta glucan, maybe this compound is one of the most studied nutraceuticals for animals. And we would like to start showing one study that studied the immunological aspect of the consumption of 1316 beta glucan from yeast. And in this study, they compared two preparations, beta glucan 1 and 2. And the main difference here is the concentration of glucans, that is 77% here and 55% here. And to balance this difference in concentration, they tested three diets: a control diet without beta-glucan, a diet with 15 milligrams per kilo of the preparation 1, and a diet with 25 milligrams per kilo of the beta-glucan 2. With this, they overcome or compensate for the lower concentration in the preparation 2. And all of this was supplied for 28 days via feed to animals. So we are observing here the effect of approximately 1 month intake of this concentration of these two beta-glucans. And what is also interesting in this experiment is that they did a comparison among different species. So they use mice, dogs, piglets, and chickens. And all of these animals receive the same dosage. And we have a huge database of beta-glucan effects in mice and even in people. And with this study, we are more comfortable now to translate this data to dogs and cats where we have much less information. So consider that the limited information we have in dogs and cats, a study like this that compared the same products in different species and showed that they act in all species, make us more confident to translate or use the information in other species to dogs and cats. And the results that they found, the first was the increase in interleukin 2. So interleukin 2 is related to the development, proliferation, and differentiation of T cells. And also it increases the natural killers and monocytes cytotoxicity. And we can see here that interleukin 2 in the serum was increased in all species, with a tendency to better increase in beta-glucan 1, but both have the same, and also in dogs, piglets, chicken, and mice. A second result, very interesting, is on peripheral monocytes and also neutrophils. And these cells was more capable to do phagocytosis. So the phagocytosis percentage is very important because this is an essential to pathogen elimination and homeostasis maintenance. So we are evaluating here the innate immunity, showing that after the intake of beta-glucan, both preparation in all species, the cells are more prepared to remove these antigens that could be a virus, bacteria, tumor cell from the body. And this is very interesting. Also, they measure an effect on antibody production that is related to humoral acquired immunity. So we saw a better, the authors saw a better response in all species. And here we have an additional treatment that is the adjuvant. So they use an adjuvant to increase immunity that is usually used. And they stimulated animal against ovalbumin just as an antigen to measure the response. And they can saw that for all species, both beta-glucan are capable to induce better humoral response in the immunity of the animals. So this is very emblematic or very studied and is the main focus on beta-glucan for many years was on immunology. However, we have other described effects of beta-glucan in animals. Other than they help immune reaction stimulation and better immune protection. Also, we have data showing that they can help on wound healing, glucose and cholesterol levels when the animal have some impaired glucose metabolism like in obesity. They stimulate myelopoiesis and also helps the response of animals to stress. And what's the characteristic of the yeast beta 1316 glucan that allowed for this huge diversity of actions in the body? And preparations of yeast cell wall is recognized for more than 60 years that induce activation and of white blood cells and better resistance to disease in people. And after several years, it was recognized that what was in these preparations of yeast that have the functional aspect was the beta-316 glucan. And this happens because of a unique molecular configuration and that this molecule have. And these characteristics of beta 1316 cannot be confounded with the configuration found in vegetables, like in oats. There is the beta 1314 that don't stimulate the immune system of the animals. And here is scheme of the molecule. So these are the sugar units, and this is the beta-1,3 linkages, and then we have also the beta-1,6 linkages. And in yeast, they are long chains. What is important to the interaction with the host of the cells of the human immune system. And here we have a scheme of the molecule. So we have glucans in bacteria, However, they are beta-1,3 linear form. In fungi, and we have here the beta-1,3 and beta-1C links, but they are short in length. And we have in yeast the beta-1,3, beta-1,6 long chains that is present in yeast. And in cereals, as I commented before, we have the beta-1,3, beta-1,6 glucans. That is linear and do not interact with the immune system. They also have some health implications, but it is related as a fiber function, soluble fiber function of these glucans from cereals. Uh, Also, one common question is why a purified beta glucan? Why not only add some yeast or yeast cell wall to my product? And this is explained because in the configuration of the yeast cell wall, glucan is in internal layers, and the external layers is composed by mannan oligosaccharides, as we show you in a minute. And the enzymes of the gastrointestinal tract of the animals are not capable to remove these external layers. And when this was ingested, it do not are exposed to the immune system, and they will not recognize that, so the animal will not be stimulated. So the active components need to be released, removing these mononucleosaccharide layers, external layers. And so these lateral chains, as I told to you, are very important for the function of the beta-1C glucan, and they need to be presented to the immune system. And also it's important to consider that if we remove this chain during the purification process, the compound will lack the effectiveness. So we need to remove the external layers, but we can't damage the beta-1,3 and 6 glucan structure. Otherwise, we can produce a compound that have no effect more. And this is why we need to test the product that we use and need to be sure that the manufacturer did a good job removing, exposing, purifying, but not damaging. In this picture, we can see that the external layers of the yeast cell wall is composed by mannan proteins. Then we have some chitins and beta-1,6, beta-1,3 glucans in the internal layer, and then the cell membrane. And all of these need to be removed without creating problems or removing these layers of beta-1,6 here. Otherwise, the molecule will lack the ability to stimulate the immune system. And how is the mechanism of action of these special sugars? So we know nowadays that monocytes, macrophages, dendritic cells, neutrophils, eosinophils, a lot of different cells of the immune system have membrane receptors that can recognize and where beta-1,3,1 glucan can be attached. So they are the CR3, dectin-1, and toll-like receptor 2. And when the beta-glucan link to this receptor, this induces a cascade of cellular events related to innate, acquired, and inflammation. So what they do is they modulate the immune system, and also they modulate inflammation. And we will show you that usually they reduce the inflammation, what is very important and can also be explored in communicating the health benefits to dogs and cats. So this is a simple list of some of the induced mechanisms, like they induce phagocytosis, as we showed first in the first experiment that we showed to you. oxidative burst that is important to kill pathogens, production of cytokines and chemokines, killing activity, reduction of inflammatory cytokines, EL6, TNF-alpha, I will exemplify in several studies in dogs and cats, T-cell activation, and so on. So they are very diverse in function. And here I can exemplify to you, this is a purified molecule of beta-1,3,6 glucan, and this is the dectin-1 receptor. When we have this linkage here, we have activation and production of nitric oxide, reactive oxygen species, cytokines, and a lot of effect that will be translated in whole body. Sorry. And here we can see in this picture that this starts in the intestine, because although beta glucan can be used intravenously, for us in diet, it's important, in nutrition, sorry, it's important that it's part of a diet. So they start all of this process in the gut, interacting with the gut immune system. But they translate this and amplify this, and they can go in different parts of the body, like tonsils, lymph nodes, and bone marrow, and they will induce a systemic effect, not only a local effect in the gut. And another example is this very recent publication 2 years back about how the fungal beta-glucan interact specifically with mononuclear phagocytes. So not all the cells, but only these. And we can see that they propose several mechanisms. And I'm not an immunologist, and this is not the idea of our study, of our presentation today, but also to let you know that we have precise information in how this type of compound work in the body. So here we have, for example, dectin-1, CR2, toll-like receptor 2, dectin-1, and depend on the situation, like where the link happens, we have here, we can see several functions in the body, like hematopoiesis, activation of phagocyte function, natural killing, phagocytosis, autophagy, oxidative burst, inflammatory mediators, production of, and protectant about reinfections, and so on. And knowing that in animal models, what we know nowadays in dogs and cats. And for the remainder of the talk with you, I will present some data that I collected, I select from the many others that have on dogs and cats specifically. And the first one that I use to exemplify possible actions and possible claims that you can use to create super premium foods supplemented with beta-glucans is this experiment in periodontal disease in cats. So the periodontal disease is very common, is a low-grade chronic inflammation in this area of the periodont of the animal. It's consequent to the plaque and a calculus accumulation. And in this situation of natural inflammation, these authors tried to see effect of beta-glucan, and they supplemented cats with 65 milligrams per day of 1,3,6 beta-glucan. One they found is an anti-inflammatory effect, reducing interleukin-1 beta and NF-kappa beta factor. And interleukin-1 is produced by activate macrophages, and it induces inflammation in the body. So also it increases the autoimmune and inflammatory states. And NF-kappa beta is a cellular response to stress, and it induces production of cytokines. It is produced under the stimulus of cytokines, free radicals, UV light, and bacterial and viral antigens. So when they found this effect reducing these components in the blood, they are showing metabolic that the animal is in reduced body inflammation. And what is most important and relevant, they follow the teeth by x-rays and they found a reduction in alveolar bone mass loss. So cats experience less aggression or less inflammation in the teeth, and do this, they have less lack of mass of bone. And this is important because it may help for a long animals to react better for this situation. And I don't know, but veterinarian knows that with time, the progression of the erosion in the bone and with a lack of the teeth, and the animal will have normal teeth to eat, and this is a big problem. And another study we did here in our laboratory, we use beaches and the neutering surgery as a model to understand the if possible effect of beta glucan. So we use 10 control beaches and 18 beaches that was neutered and received 0.1% of beta glucan, or the same this is the percentage of the diet, or the same dosage but in capsules. And the control beach was neutered also, but do not receive any beta glucan in the period. And what we found is after 14 days of neutering, the neutering is immunosuppressive processing because of the stress and the anesthesia. And we can see here that the leukocyte reduced, but coming back, but the return was faster and higher in the both supplemented animals, showing that beta glucan prepare the animal for this stress situation. Also, we saw an increase, especially in the beta-glucan fed by food, in T cytotoxic lymphocyte, that is the cells responsible to killing foreign cells or antigens. And this both here we see a reduction induced by surgery and a return to normal level. But when animals receive beta glucan, they have higher levels, so they are more prepared. The cellular immunity of this animal is better. And also we measure in these beaches a difference in C-reactive protein. So C-reactive protein is produced by the liver on the situation of inflammation or infection. And after the neutering, the surgery, we have this increase. This is the day after, not enough time to increase C-reactive protein, but 14 days after we have this increase. But the increase was higher in the control animals. So we can understand that in this moment, 14 days after surgery, these control dogs are more with higher inflammation and higher stress. And these animals are more controlled, or we fasted the return to basal, the wound healing, or in other process in the body that makes the animal healthier sooner when fed beta glucan. Another interesting example for you is on puppies and immunology. So in this paper, the authors described the effects of the oral supplementation of 1,316 beta glucan in puppies. So they used a group of 15 puppies that fed a controlled diet without supplementation, and 15 puppies that receive orally 4 milligrams per kilo of body weight of beta glucan. And they are vaccinated against temper, leptospirosis, rabies, and several other diseases commonly used to vaccinate puppies. And what they found here, we can see that after the start of the intake of the products, they differentiate in the phagocytic activity index. We showed this in the first experiment, and we show that beta glucan prepare the innate response, and with this we have better phagocytosis. And after 28 days, 1 month approximately of the intake of the compound, the supplemented pups have better phagocytosis, so they're more capable to develop an immune response against an infection. And also they produce more antibodies. So here is the antibodies against parvovirus, and we can see that supplemented animals started to produce sooner and produce higher amounts of antibodies than the control puppies that were not supplemented with beta glucans. Also here we have neutralizing antibody against rabies, and we can see here is considered the point where the animal is protected, and we can see that the supplemented group are protected sooner, 15 days, and the animals that did not receive beta glucan delayed some days more, and also animals develop a higher titer of these antibodies, so they might be more protected against rabies because of the supplementation on the basal diet that they have of beta glucans. And this is another interesting experiment of Dr. Bainen, where the authors evaluated dogs with atopy. So atopy is a common skin disease used when animals develop allergy against the environment or compounds in the environment. So they have a lot of skin problems. And they selected 16 atopic dogs for a control diet. In fact, I guess this is 15. I typed wrong, and 15 atopic dogs in a diet supplemented with 0.8 grams of beta-316 glucan per kilo. And this was a double-blind placebo-controlled study. And here they have, after the intake of the diet, some indices like pruritus, redness, scaling thickness, stripping. And when they combine all of these previous index in a total index, they found an interesting effect, reducing this chronic inflammation or the symptoms of the atopic dog because they are eating beta glucan. And this is very interesting because it was a placebo controlled and evaluated the situation in a natural condition. And as we showed you before, beta glucan interfere in the inflammation process, and they reduce pro-inflammatory cytokines, and this might help a better structured skin. No, the animal have less side effects or less pruritus, redness, and this. Dr. Bain and groups also did another interesting experiment with another model of chronic inflammation, that is osteoarthritis. So they selected 23 dogs with naturally acquired osteoarthritis as a control, and another similar paired group with osteoarthritis eating a diet with 0.8 grams of beta glucan per kilo. And what they found was interesting because the activity level of the animal increased 80% in the beta glucan group. The stiffness increased 15%. Swelling reduced 4%. This was not significant. But lameness reduced 10%, and this was significant. And pain also reduced, and this was also significant. So we saw here that this interference in the immune system of the beta glucan may also help not only skin, but here arthritis. And this is examples of situations, natural situations that dogs and cats have in their lives. And a diet planned for that can help the quality of life of these animals. And this is something that you might communicate if you do a proper evaluation and confirmation of this. And this is important because we don't need only to add that in a diet. It's important to plan the diet, entire diet for that situation, that they have a proper balance of nutrients, that have very safe situation, so they are safe, don't have any kinds of problems, as I commented before. And also that with the nutraceutical, they was evaluated and showed this benefit. This is very interesting. Uh, Another study that was very good for me is the effect of these compounds on inflammatory bowel disease. So inflammatory bowel disease is common nowadays, is a chronic disease with difficult management, needing hypoallergenic diet, anti-inflammatory drugs, and antibiotics. And this research group compared some immunomodulators. So they used 7 dogs as a control, 7 dogs supplemented with 7 milligrams of beta glucan per kilo of body weight. And also they have group 2 and 3 that we can see here, that is beta hydroxy, beta methylbutyrate, or levamisole as immunomodulators that they experience. And the first experiment, the first result that I show you is the CBDI. This is an index validated by the American Association of Animal Veterinary, Small Animal Practitioners. And this validated score was similar in the start of the study, but after the intake of the diet supplemented with beta glucan, we can see that this score comes to almost 1, less than 1. And also the compound 2 and 3 also have some help, but here not statistically. And control animals experienced in the period an increase in the index, the CBDI index. So showing that the presence in the gut of beta glucan help reduce inflammation. And what's the symptom? Here we have the recurrence percentage, animals that experience again symptoms of the disease. The symptoms are vomiting, abdominal pain, diarrhea. And none of the animals treated with beta glucan experience symptoms again. And all of the control dogs experience symptoms again. So this is very different, very emblematic, this response. And they evaluated the mucosal score by endoscopy. So they collected at the start and at the end of the experiment samples of the gut mucosa. And here I'm showing you only the group 1. And we can see that all the 7 dogs of group 1 have some grade of inflammation, moderate inflammation in duodenal evaluation. And after the immunomodulation or after the intake of beta glucan, they are in or normal. So just feel very mild reaction, inflammation or normal. And this confirm that the lack of symptom was because beta glucan reduced intestinal inflammation in these animals. Biochemically, they also observed a reduction in interleukin 6 and increase in interleukin 10. So interleukin 6 is a pro-inflammatory, interleukin 10 is anti-inflammatory. It helps balance inflammation and not overstimulate inflammation and immunity. So it's very interesting, this finding that they increase these anti-inflammatory cytokines and they show a better inflammation of the mucosa, and these are very linked and can explain the lack of symptoms and the better quality of life of these dogs. And going to the last experiment that I selected to show you is about the use of beta glucan in obese dogs. So we know that obesity is a situation of chronic low-grade inflammation, and obesity is well known that also induce alteration in glucose metabolism with higher glucose and higher insulin secretion in the postprandial period. And these authors here from Brazil, they select a group of 7 obese dogs and 7 non-obese dogs, and they just fed a diet, a controlled diet for both the same diet in a level to maintain body weight. We are not telling about loss of body weight. They maintained the body weight, and after 15 days of eating this common diet, this basal diet, they did an intravenous glucose tolerance test. Then they fed again the control group, the obese animals, with a 0.5% of beta glucan in the diet. So they supplemented the diet with this amount of beta glucan and fed only the obese dogs for a period of more of 100 days. So a long period. And then we will see again in the next slides the basal results of the obese and non-obese, and the final results of obese after eat the beta-glucan supplemented diet. And they found that the basal glucose was higher in obese and lower in the non-obese and obese after eat the diet with beta-glucan. It's important to consider these obese dogs did not lose body weight. They maintain the obese condition. The beta glucan induces some change that reduces glucose. When you look at the basal insulin, very high as expected in obese animals, lower as expected in the control non-obese animals, but it reduced after the intake of the beta glucan supplemented diet. And also the mean value of glucose follow the same pattern. And other interesting outcome was on cholesterol and triglycerides, where they found higher levels, even higher than the normal range in the obese animals. And then normal values for non-obese dogs. But after the intake of the diet supplemented with beta-glucan, the obese animals presented lower cholesterol and lower triglycerides, lower values than they have when obese and not supplemented. And this is interesting to observe and to understand this. Maybe we can look at the results on TNF-alpha and GLP-1. So GLP-1 or glucagon-like peptide-1 is produced by neuroendocrine cells of the gut and is stimulated by several compounds like fermentable fiber and like here in beta-glucan. And GLP-1 have anorexigenic effects that may favor body weight loss. It's not the case of this study here, but it stimulates insulin secretion and mainly peripheral insulin action. So it increases the sensibility of the peripheral tissues against insulin and do this have a hypoglycemic effect. It's so important, so strong that it's tried as a drug for people with type 2 diabetes mellitus. So the increase observed here, we can see that the basal are similar in both groups, but after the intake of the diet supplemented with beta-glucan, obese dogs experience a very high, much higher level in serum level of GLP-1. And also an important reduction in TNF-alpha. There is a pro-inflammatory cytokine. So it reduces from 9.5 to 0.9. Sorry, 4.9 to 0.9. And TNF-alpha is a pro-inflammatory cytokine, mainly secreted by macrophages. During obesity condition, it is secreted probably by macrophages in adipose tissue, and it is one of the responsible to keep a low grade in chronic inflammation in obese animals. And also high levels of TNF-alpha is associated with insulin resistance. So the reduction in TNF-alpha together with the increase in GLP-1 might explain these alterations in insulin, glucose, and cholesterol levels of these animals. So coming to conclusion, a super premium pet food, when we look at, owners expect a product that, besides superior nutrition and high safety standards, might induce additional benefits to good health. And super premium products must have formulas very technically established based on complete and balanced nutrition, adequate standards on food safety, but also health claims that is scientifically proven based on scientific publications in animals. And considering all of this, the beta-3 and 1C glucan derived from the yeast cell wall is the most studied nutraceutical. As I showed you, they have more 30,000 studies. It's a huge amount of information about it. And they have high possibility to explore, to modulate inflammation, like we saw in periodontal disease, arthritis, atopy, skin atopy, and inflammatory bowel disease. So this compound may help to reduce in some situation the inflammation. And also it's proven to create better immune balance and immune capability. And we saw studies in mouse, chicken, rats, dogs, and puppies showing the better title, better phagocytosis activity, better killing activity. And also in this last study, we saw that it may also help to control glucose and cholesterol levels in the blood. So we can see that with unique molecule, it's important, it's possible to create many different claims depending on the formulation or the product that you are trying to develop. So that is what we prepared to discuss with you today. Hope you enjoy it and hope you consider this information useful for you and your business. And again, thanks a lot, Biorigin, to invite me to be here and speak to you today.
Thank you, Professor. Now we are starting for the questions. The first one is, how are you sure that you do not overstimulate IL-2, which can be bad at long term?
Hello, good afternoon to everyone. And this is a good point. And interleukin-1 can have bad effect, but also good effects, because when you look at in chemotherapy, cancer therapy, it's one of important drug to use against cancer in several organisms, including it is used in therapy for people with cancer. And this is because of the preparation that they have for the immune system as we show it. And overstimulation of the immune system is also a big concern when we look at the immunomodulators. But I think that it's not the case of beta-glucan, because when we look at, for example, the studies on arthritis, skin disease, inflammatory bowel disease, when we have the disease is an overinflammation and maybe autoimmunity. And this disease can also be controlled by the beta-glucans, as we saw in the results of their studies. And also, the most important pro-inflammatory cytokines like TNF-alpha and interleukin-6 usually are decreased when the animal receives these kind of compounds. So looking on that, the balance that we saw in some disease regarding inflammation or controlled inflammation, I think that it more prepared the immune system than just put every cells of the body in high level of activity that for sure that's not good. So it's my point of view.
The second question, is there an evidence of impact of degree of branching on immunomodulation? If immune system will not be able to recognize beta-glucan, if there is a layer of mannan, what about all its cell walls products in the markets? Most of them contain both components insoluble. Are they still effective immunomodulators?
This is a good point and a little complicated because we have many pathways to finally achieve stimulation or a preparation of the immune system. By coincidence, to make easier to give an answer, a possible answer for this question, I started showing that we did for Biorigin, I study about the solubility of monoligosaccharides. And we saw that this product was different, and they are fermented, induced the production of beta butyrate in the gut, and through this pathway, it also stimulates the immune system. And it's very important because to understand this, to remember that 70% of the immune cells in the body are in the gut mucosal, is the gut-associated lymphoid tissue. And when we change the environment in the gut, and when we reduce the inflammation in the gut, as we saw reducing TNF-alpha, for example, with monoligosaccharides, we end having a better stimulation of the immune system. We can see responses in vaccinations. In our particular study, we found better phagocytosis index in the monocytes, peripheral monocytes. And so by different pathways, they end with the same. But here, when we look at the beta-glucan, we are talking in a very different pathway. We are not telling about fermentation or short-chain fatty acid production. We are telling about a direct recognition of the molecule by the receptors that is well described, surface receptors in immunological cells. And when these receptors are stimulated by the link between the beta-glucan and the membrane of the cell, they develop different response, cellular response. And we illustrated that in 2 slides, for example, and we have plenty of studies showing that. So for a product doing stimulation in the animal, stimulation is not maybe the better word, so a preparation, because overstimulation, we have the first question, it is not very nice. So to the immune system be prepared and be more strong when after the intake of beta-glucan, we are telling about the direct recognition of the molecule by dendritic cells, macrophages, as we showed. And if they are uncovered by a layer of monoligosaccharides, you do not have this effect. You maybe have an effect, but it's by the other pathway, the mannan pathway, fermentation, and a prebiotic effect. So it might confound in the end what's happening with the animal.
Yeah, and just to add a comment, we have some difference, some evidences based on the difference of beta-glucans obtained from different sources. For example, we have, I can share with you later, but we have difference comparing the beta-glucans from bacteria, from cereals, and the most efficient for, as Aulus told you in the webinar, to stimulate the immune system. The most used or the most efficient is the beta-glucan 1316. So that's different. The structure will be different of the molecule, will be different from the just 13, like a linear structure, or 1314, okay, like inode or other source of beta-glucan.
And another small comment is about dosages. We can see that beta-glucan works in small dosages than common prebiotics, because prebiotics need to be in enough amount to induce change in microbiota. And they only works by feed, by foods. And beta-glucan can work if infused in an animal, when used as vaccine adjuvants. We did trials here, putting them in cell cultures, and the cells recognize them and they stimulate the cells. All of this will not happen if they are under a layer of mononucleosaccharides.
And the last question, Professor, what is your opinion about the association of a source of mannan protein and a purified beta-glucan?
Maybe mononucleosaccharides, I don't know. Because if you associate mononucleosaccharide, there is a prebiotic with beta-glucan, you can do this doing a good separation of the cell membrane components, and you might have a nice combination, but that need to be tested because both can have similar end results by different pathways, and they using the mononucleosaccharide, you can have induce a better environment in the gut. You can modulate microbiota metabolism activity, and this can reduce gut inflammation and prepare the immune system by some pathways. And if you combine this with beta-glucan, it is possible that you have a different or by a different mechanisms, as I show you, by direct interaction with the immune cells, you can also have the preparation of the immune system. And I'm not aware about a study that combined the two and see if they are additive or if they potentiate each other, but that might be an interesting proposal for a research.
Thank you. Thank you for all. Thaila, would you like to say something?
I think we are finishing our webinar. Aulus, would you like to say some words first and then I finalize the webinar?
I hope the information we share was useful to you and stimulate your curiosity to search at the websites and look at the scientific information that is huge about these compounds. And thank you, Biorigin, to invite me. It was very good to be here with you this afternoon.
Okay, thank you very much, Aulus, on the behalf of Biorigin. So I'd like to say thank you and to say that's for us, it's an honor to have you here. I would like also to thank you all the audience. Thank you very much for your questions, for your participation. And if you still having some doubts about, please get in contact with us that we are going to be very glad to answer the questions with you and to talk a little bit more about the beta-glucans or the soluble bananas. Okay, thank you all of you and have a nice day. Bye-bye.